All right, since it's 3 o'clock, I'm going to start. Okay, so I'm giving the next two lectures, lecture 14 and 15, alignment and visualization. But before I talk about the material that I prepared, a lot of the material that I prepared is taken with permission from Dan DePledge's sequencing informatics course, and also from work from Aaron Quinlan's lab. And here I've also assembled some helpful links if you want to look further into some of the things that we're talking about today. So what I'm planning to do is for lecture 14, it's going to be more lecture-based and a little less hands-on, so we can just think about what alignment is and just start thinking about the different parameters and things we have to think about when we do the alignment. And then on Thursday, we'll actually run the alignment and explore visualization options. Okay. So this is a workflow of what's been done before we touch our FASTQ files. We have our cell, and this is an example of RNA sequencing. So we have our cell, we have our DNA, and someone spent a lot of time assembling a reference genome or transcriptome, and they made it available for all of us to use. And then on the bench, someone has extracted mRNA or whatever you're going to sequence, and then that is sequenced, and you get your output FASTQ format files. And those FASTQ format files are what you worked with Lisa last week. So what we're doing is we're taking these the reads from the FASTQ format files, and we have to align them. We have to map them, so find their space where they should go on the reference, align them, and then in next week, Igor will teach you how to quantify within specific regions of your reference how many reads align to that region. But today, we're focused on finding the places for these reads on our reference. And there are a few caveats to aligning this way. You can see that we're really basing our results on the reference alignment. So if you are working with something where you have large insertions, or for some reason an organism without a reference genome, you're not going to be able to use alignment methods, and so you should look into assembly approaches. But that's outside the scope of today's lecture. And so you've seen this workflow from Dan in his first lecture, but the standard workflow is you get your raw sequencing reads, you do quality control, you do quality trimming. Here is Lisa's week, and then this section here is going to be what we go over in my week, and then this here, one of these branches here is what you'll go over with Igor. So you saw on the last slide, we have to find our reference genome, our reference annotations, and figure out which aligner, which program we're going to use. So there are two main ways you can get a reference genome. The first is you can go to Ensemble, and I have made screenshots and put in boxes your step-by-step path to get to the file. So if you're in Ensemble and you want to get the human genome, you're going to click the human. Then you go over and click download DNA sequences. You click that, and there's a whole list of different FASTA files that you can use as your reference genome. And if you're using Ensemble, the file that you're looking for is this one here, Homo sapiens, the primary assembly. If you're interested in learning about all those other files, they have a website here that walks you through the different files that they have there. Now you have to also get your reference annotations, and you're going to get those from the gene annotation side. So you're going to get them from this download GTF link. And it takes you to here. And the file that you want is Homo sapiens, this GRCH38.96 chromosome GTF.gd file. Now, if you're going to be downloading things from the UCSC genome browser, which gets a little convoluted, you're going to go to Downloads, Human, and then you're going to follow their links. So you're going to click Genome Sequences. You're going to have to scroll down this huge wall of text. You're going to go to the analysis set. Their analysis set, they say, is no longer being updated. However, where their analysis set is being updated is from this link right here. So if you copy and paste that link into your browser, it'll take you to this web page here. And from that web page, you're going to select just analysis set FNA. FNA is the same thing as a FASTA format file. Now, if you're obtaining your reference annotations, you go down the same route, but instead of clicking analysis set, you're going to click on genes. And then here, what's great about the UCSC genome browser is that they give you different annotations. So there are annotations from Ensemble, from NCBI RefSeq, from RefGene, so you can choose what annotation you're going to use. So you download one of those. Now, if we have time later today, we're going to download those programmatically. And if not, we'll do it on Thursday. But usually you don't just click on the file and download it to your computer because the reference genome is really, really big, and you don't want to download it to your computer just to upload it back to Big Purple. We'll just directly from Big Purple download it from the web. Okay. Now, if you can't find the references that you need from those two sites, you can go to Ensemble Genomes, and you can try to look for your reference genome or transcriptome here. Okay, so that's where you look for your references. Now you have to choose an aligner, and there are a lot of different aligners out. Here's a plot showing on the x-axis is time, and here we're showing when different aligners came to be. And you can see there's a lot of aligners. Does each user have to download a standard reference to Big Purple, or is there a shared directory / path? I'm not sure if there's a shared directory for everyone. We're gonna download standard references to our own folders as part of the class. So the genomes for Ensemble and UCSC should be the same. Like the references shouldn't be different. Oh, Sana says that there's a module that helps you get those standard references on Big Purple. I didn't know that. But what's great about UCSC is the GTF files that have different reference annotations for different databases like RefGene versus Ensemble. Okay, so you might be asking, why are there so many programs to do aligning? Like, you can just search things on Google and things pop up. So why do we have to have special algorithms to align sequences to the genome? Why can't we just, Control F? Well, the thing is, mistakes are propagated through to the FASTA files at every step. So the samples might be degraded. Something might happen during the PCR amplification, or when you're sequencing, the machine just might sequence wrong. So you don't know for sure that the read that you're looking at is error-free. And for sure, it's not error-free. And when you go try to match that read to the genome, and you just use a Control F function, you're going to lose a lot of your reads because a lot of them aren't going to match perfectly. Okay, and then just two different things. Mapping just means finding the best location of the sequence on the reference. And then aligning, it's going actually base by base, aligning that read to the reference. Okay, now there are different types of aligners. If you're doing RNA sequencing and you want to use a splice-aware aligner, like BBMap, Minimap2, Bowtie, or STAR, and that's because these aligners are capable of splitting one read across long distances. So basically splitting up your read to only map to the exons, like you can see in the schematic I made down here. And something to keep in mind is that these splice-aware aligners usually have different flags to indicate whether the data is from RNA or DNA. So if you choose RNA, then it's going to go the splice-aware route, but if you choose DNA, it's not. And those flags, it's important when you're using a new aligner to read through basically the instruction manual, because there's a lot of parameters that can control the sensitivity of the splice detection, and we're going to see other flags that can affect the quality of your output. Okay, and then I think Dan went over this in his lecture, short versus long reads. A lot of what we're going to be working with is short reads. I think mostly most of RNA sequencing still is short reads because it's higher coverage, but it can't resolve repetitive regions. So these are technologies like Illumina and Ion Torrent, whereas long reads, you can get more information per read, but you know, you'll probably get fewer reads, and these technologies are things like Nanopore and PacBio. So back to choosing an aligner. If we're looking at short read alignment, these are Dan's choice tools, and I also agree. I've never really used Context Mapper. I've used BBMap and STAR and Bowtie. And then for long read alignment, he suggests Minimap2 if you're doing long reads. Okay, so just the broad strokes of short read alignment. You have to align a lot of short sequences to a huge reference sequence with the expectation that you're going to align it very well. And so as your read gets longer and longer, the number of mismatches increases. And also, you want to be able to rank your alignments and align it as well as you can to the reference genome. So the first thing that you do is you build an index for the reference sequence. And we have to build an index for the reference sequence just for computational efficiency. People always say that, like, if you stretch out your DNA is so long it can go for I don't know, forever and forever. I forget how long it can go. But if you think about the length of DNA and you're just wrapping it around and putting it in a simple text file, that text file is going to be huge and really unmanageable to work with. So different approaches for indexing. Here are two different approaches for indexing. Help us really help with computational efficiency. And I know Dan went over this as well, but just as another refresher, here, after you've done your initial prep, you have your cDNA fragment, and this is after fragmentation. So this isn't the full length of an mRNA. You have your adapters here, and if you're doing single end reads, you have a read going from one end of the read, but if you have paired ends, you're going from two ends. And I bring this up because this fragment fragment length here is important in this slide. Okay, so the best case scenario is that your read aligns to the reference and aligns to one region of the reference. Usually what programs do is they select one part of your read to just find a portion for that initial seed, and then it'll see whether the rest of the read matches to the reference for efficiency. So here I'm showing a best case scenario. I'm showing these green lines as indications of alignments, and here we're having paired end reads, and we can see that the paired end reads are about the fragment length size. So that is good. Okay, but like I said, the reads can have a lot of errors in them propagated through. So your read might not match exactly to the reference genome. So here I have in red regions where the read doesn't map to the reference. But sometimes, you know, it almost matches, like there's one mismatch or two mismatches. And this is where the different flags and the different parameters that you use when you run your alignment step really matter. So you can say you're allowed to have 2 mismatches and you can be aligned to this position, or you can increase the number of mismatches. However, as you increase the number of mismatches, you know, the number of mapped regions increases, but so does the incorrectly mapped regions. So you have to balance those 2 forces. Okay, and there's also a possibility for multi-mapping reads. So if a single read can map to many regions of the genome, that's what a multi-mapping read is. So here I'm showing the same read is mapped to many places. And again, this is where the flags that you use, the parameters that you use really matter. So you can choose to ignore ignore mappings that appear more than whatever n number of times. As you increase that n, you'll get more noise in the data. So that's another thing that you have to think about when you play with these parameters. And these multi-mapping reads are usually in regions that are repeated regions in the genome. Yes, I did not put the slides up on the course website yet. I apologize. And Nicholas says, what does the seed portion refer to? The seed portion refers to just a lot of the times programs just use a small portion of the mRNA to search around to see where it fits, and then it'll look at the rest of the portion just for computational efficiency. And Kate asks, what is a good cutoff for n times for multi-mapping reads? I'm not sure. I think it depends heavily on your task. And honestly, I'd have to do more research on that for a good cutoff for n times, so I can answer that on the Stack Overflow later. Okay, and then we have, as a recap, we have our primary alignments, which means that this read aligns to one place in the reference, and this is the best outcome, and this is the most common outcome. So this is a good alignment. Our reads align very well. They're within the maximum fragment size, and they're uniquely mapped. However, we have our secondary alignment. So if multiple primary alignments occur, you have to choose which one will be a primary alignment versus a secondary alignment, and sometimes those are just random. So if this is if this is the case, then the algorithm will just choose one of these randomly to be the primary alignment. And then in terms of this term called supplementary alignment, this is what happens when you have maybe structural variation in your RNA. So one part of the read, so this is the same read here that I'm showing, but the first half aligns to one part of the reference, and then the second half aligns to somewhere completely different, a little far away from the read. So this is what would be called a supplementary alignment. And I think you guys went over with Dan before the SAM format. So I'm not going to go over the SAM format, just that once you have your SAM format, you have to convert it usually to the BAM format, which is the binary version of the SAM format, and it saves a lot of space, and it's used as input for a lot of the visualization tools. And on Thursday, we're going to go through some handy functions and commands using SAM tools, but SAM tools is really what's helpful in manipulating these SAM and BAM files. So if we look at the SAM format, we have our flags here, which tell us about some quality and type of the alignment. And here we're going to talk about our cigar strings. So here the cigar strings tell us really how that read aligns to the genome. So before I get to the cigar strings, Dan showed a slide about the mapping quality with BWA, for example, and he had in here in red that mapping quality scales and scores are dependent upon software. And I want to show you that that's really true. So here, this is the same slide from Dan. We have our BWA mapping quality, where a read aligns when a read aligns many places, it's 0. And when a read aligns to only one place, it's 37. But for star, when a read aligns to one place, like a uniquely mapping read, it's 255. And 0 is when it maps to a lot of different places. So it's important when you're filtering your alignments after you align. If you're only looking for regions that are uniquely mapped, if you're using BWA or star, the flag, the number that you're going to be filtering for is going to be different. Okay, and then moving on to cigar strings. Cigar strings stand for Compact Idiosyncratic Gapped Alignment Report. And basically it tells you how this read aligns to the reference. So I'm going to show you an example I found from Galaxy. Here is our read, here is the reference. And if we walk through the cigar string and we look back to however you go back. Okay, so we're just going to look at these first few operators because these last few ones get more complicated. So the first one, M, means that there's an alignment match. I means insertion against the reference, and D means deletion against the reference. So if we go back to an example, we can see here we have a match. So that's 1M. Here we can see that there's an insertion in the read. So that's 2I, so insertion against the reference. We have a set of 4 matches, so that's 4M. Then we have a deletion against the reference. So here we don't have the G where the reference does, so that's 1D. And then we have 3 matches. And that's how you read a cigar string. And if you want to go through these other examples, I've put them on the slides. And then lastly, you can get duplicate reads from your library prep protocol during the PCR amplification step. If you have not enough starting material and you over amplify, you can get the too much replication of a certain read. And the consensus or the rule of thumb is for RNA-seq, you don't remove the duplicates, but for things like variant calling, you would remove the duplicates. And here, when I was looking into this, I found these forums on BioStar and Seek Answers to answer the question and discuss why we remove or don't remove duplicates. But basically, the idea is that although yes, you can get duplicate reads from PCR for RNA sequencing, you don't know for sure. There's no way to know for sure that a duplicate read is from a PCR amplification error. It might just be that you quantified by chance to the same exact regions and have two of the same reads. So for RNA-seq, you don't remove duplicates, but for variant calling, you do. And the way you can mark and remove duplicates is using this tool, this package from the Broad Institute called Picard. And you would use these functions, mark duplicates, using a BAM file or a SAM file. Okay, so we got through that in 30 minutes, so we have time to get the data from the internet. Okay, so if you're going to be running a lot of RNA sequencing, there's a tool that Igor Dogolev in my lab has created, and I think he's going to go over that next week, but I just wanted to bring it up for people to check out in the meantime. Okay. Okay, so now to the hands-on part. We're going to move to Big Purple. So let's see. Let's start from scratch. Okay, so now I'm on my terminal. Okay, and we're all going to sign in to Big Purple. So that's SSH, my KID. Okay, so I've logged into Big Purple. Right now, I'm going to srun. We can't see my terminal. Interesting. I don't know how to share my terminal.
Yep.
Now, you can see my terminal?
Mm-hmm.
You can see my split screen?
Yes.
Okay, perfect. Thank you. Okay, so we're in my terminal. And just before we do anything to cover the bases, we're gonna do an srun. And so I apologize, I didn't get you guys this, this code beforehand, but I can't even copy and paste, so we're gonna type it in together. Srun. And I can't see what I'm typing. Whatever. Okay, so as a reminder, this srun, we're logging on to a node that's not a login node. So this is one of the compute nodes. I'm asking for 4 gigs of memory. I said, give it to me for 2 hours. And hopefully I'll get the resources quickly. All right, I'm gonna get it 30 seconds and then ask for way less. All right, it's too busy and I don't want to wait. 1 hour. All right, it looks like, let's see why the cluster is so busy and I can't get a small job. All right, well, since it's a hassle and we're only going to be copying stuff, don't do this in real life. I'm just going to give up on the srun for now. Okay, so we're going to move to the directory, the class directory. So the class directory we're moving is cd gpfs data courses bi20 here. Okay, so this is what you should see in the course directory. And I've put the file that we're working with within the alignment folder. So you should be able to cd into alignment, and if you ls, this is what's within the alignment folder. We're going to move into scripts. And there's a lot of stuff in here because I was preparing for Thursday, but all we need is this file here. Now I made it so that you can't edit this file within this directory. So what you need to do is you need to copy this into your home directory or whatever directory that you can copy it to. And an example of copying it to your home directory is doing this. So CP, you're going to give the path to the file that you're copying. So since we're in the scripts directory, the path to downloadreferences.sh is just downloadreferences.sh. If you're not in the scripts directory, the path, wow, I can't see what I'm, is the absolute path, so here. So now I'm pointing to the downloadreferences.sh file, and now I have to give it a path where to copy it to. So I'm going to copy it to my home directory, gpfs home. Okay, so this should copy that file to my home directory. And that's not how you spell it. I can't see. Thank you. Okay, so now if I check, I like having 2 terminal windows open so that you can go from one place to another. Oh, here it is. I didn't see it. It's in green. Download references.sh. And if we look, please pardon my dirty directory. this is where I am now. So we moved the download references.sh into a different folder. Okay, now if we look within download references.sh, we see our sbatch header. So I made this an sbatch script. So you can run download references.sh using sbatch. But we're not going to run this file. This is an example for you guys to work with. You can see here we are using a wget function. And I think you guys have used wget before with Angelina maybe, but what you need for a wget here, you can see in the slides, is you use wget, you use the flag -o, you give it the path, the output path, so your big purple path, and then the internet path. So I gave you the internet path here, but here in this output path, you guys can't write here because I didn't give you the permission to write here. So you have to change this output path to your own output. So whether that be your home directory or wherever, your lab directory. So let's pretend that I also don't have access. I'm going to create a new directory called bi20 test. So now I have a new directory called bi20 test here. And you can move into bi20 test doing cd bi20 test, and now here I am. All right, so let's do this all over again, but I'm going to copy into. Okay, so I have my file in bi20 test. I'm going to, now I have to change the path, the big purple path within download references to this path. So I think Angelina showed you Vim, so we'll use Vim. But also Lisa did Nano. So I'm gonna do Vim first and then I'll do Nano. So Vim, download references, enter, okay? To edit in Vim, you need to press I to edit. So here I typed I, okay? Now I can edit stuff. Here I'm going to change the path to where I'm going to move. I'm going to save this data. So it's GPFS home, AY1392, BI20 test. So what this line of code here does is it takes this file, this fna.gz file from the internet, and it's going to deposit it to this file here. And I'm not actually going to run that on the head node. That would be bad. Okay, so the thing is, you can go through and change the paths for every one of these. So you can see that the next thing I did was, you see this FNA has a .gz. You have to change the .gz. You have to unzip it basically. So you're going to use gunzip, this .gz here. You use gunzip to unzip the file. And then we do the same thing to get the GTF files from UCSC. So I got both the ref gene GTF and then also the ensemble gene for GTF. So you can change all the paths here to the relevant paths for you, but I want to show you okay, so to exit, I pressed ESC and then did semicolon, WQ, and then the exclamation point, and I'm out. So if you're a Nano person, you do this using Nano, download references. You can just edit whenever you want. So let's edit this one. Oops. Okay, so my path is gpfs home, my KID, and I made a new directory called bi20 test. Okay, and then to save, it's Control C, I think, and then enter. That's not it. Control X, sorry. Control X, and then you say yes. And then you have saved it, and we can see our changes in our file. Okay, but you can see that's really annoying if we have to copy and change the paths every single time we do this. So I created another file in the courses B20, BI20 alignment scripts directory. And this file is called download references param input.sh. So let's copy that into our home or whatever directories that we're working with. So again, that's CP. I'm going to give it the path to the file I want to move. Now I need it to give it the path of where I want the file to go. So that's gpfs home AY1392 BI20 test. Okay. And then I have another tab open. So just, this is where I am. And I've moved that file to this folder successfully. Okay. Now let's look at this file. So there's something new in this file. This file is creating a variable from an input. And that's what a lot of the pipelines that, if you use pipelines on command line, a lot of those pipelines use this type of variable input. So what this $1 means is that this out path variable is being set to the first input of the file. So what this would mean and how you would use this is, let's remember our file name. And you're gonna, the way you would use this is to sbatch, but we're not gonna sbatch this right now. And then you would input the out path. So instead of having to go in and go through and delete and paste and delete and paste every single path, I created that output path. I made that output path a variable. So here we're setting this output path variable as an input. So before you were just doing sbatch and then your file, but now if you want to put an input, you have to do sbatch, your file, and then space, and then the path. So my path here is gpfs bi20 underscore test. And that is how I give the input to the script. But let's go back to the script before we run anything. And let's look at how bash deals with parameters. So I said this number 1 means that the first input is your out path. If you want to have multiple inputs, let's say, I don't know, you wanted your first file to go to one path and then your second file to go to another path. So you want your first FASTA format references to go to one directory, but you want your GTF files to go to another directory. You can put 2 paths as input into this code. And instead of just out path equals $1, you're going to have out path 1 equals $1, and then enter out path 2 equals $2. when you play around with bash scripts, there's something that everyone gets hung up on at the beginning. It's that you can't have spaces when you're setting things equal to something else. So back in when we were doing R, you could have x space equals space 2. But if you're doing that in bash, you can't have spaces. Once you have a space in bash, it thinks that you're done, and the space, the thing after the space, is your input. So here you can see we have no spaces. And to call, to use this variable, you don't just put out path like we did in R. You have to specify that this thing is a variable, and you can do this using $, these brackets with your variable name within. So here I changed the out path here, 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 all throughout. So now we're not copying and pasting and changing the paths everywhere, because oftentimes you forget one line and it messes everything up. Okay, so let's see if this works. So I'm going to do, okay, so right now I have access to execute it because it's green. Sorry, that green is just for my setup. I'm not sure what everyone else's is, but if you do LL, it'll tell you what permission you have for that file. So if you only have read and write, then you can't execute the file. But since I have read, write, execute, I can. If you don't have read, write, execute for some reason, you can change the permissions by doing chmod, which I think, honestly cannot see what I'm typing, chmod plus x, and then the file that you want to give yourself executable permissions to. So like this, you don't need a space. I honestly cannot see. Start again. Plus ch plus x. Okay, and that doesn't change anything for me because I always had the x, but if you didn't have the x, that's how you give yourself the x. Okay, and now we're gonna use sbatch. And then, wait, let's check one more thing before we sbatch, because I don't. Okay, so here it says in this sbatch header, we're gonna have, it'll email us where our, how our job is doing. So if it ends or fails, it'll email us, but we need to input our KIDs. So let's input our KIDs now. I'll use nano because it's probably easier for everyone to use. Okay, so my KID here is AY1392. And we're going to control X. Yes, I want to save, enter. Let's look, see if it changed. It did. All right, now the last thing we're going to do is to sbatch this file. So just sbatch the name of the file and then the output path. Dawn asks, so without chmod plus x, we can't run sh file a few days ago. Okay, so if you LL and you have executable permissions, so let's, yeah, you most likely have those permissions, but I wasn't sure if you had those permissions or if only I had those permissions. So if you can run it, then you're good. That means that you had the permissions, but if you don't have it and say you created an sh file on your own, it's not going to automatically give you executable permissions. You have to give it executable permissions. Okay, and let's, whoops. give it a path, GPFS, home, BI20 test. Okay, and my job is submitted. And then I think the cluster is really busy, so maybe we won't be able to see it run. Oh, sorry, that's SQ. Did I spell it right? S. Okay, so I am checking to see how the job is doing, but it's still in the queue, even though it's a small job, but it will run soon, and it should download the references to this BI20_test directory. Okay, so in the last thing I'm going to do is I'm going to go back to the courses, alignments, scripts directory here, PWD. This is another tab of mine that I like keeping open, just so I don't have to change directories and type everything out. I usually have like 3 tabs open. Okay. Okay, so I moved back just to this path here, GPFS data courses BI20 alignment, and then I put the things that were downloading in this references path. Yes. As Lisa said, don't run wget on the command on the login node. Please submit it as a job or go interactive node. But since the cluster is busy and I can't get a quick srun, we've created an sbatch, so the sbatch should do, but don't wget on the login node. And so this should be your end output minus a star alignment thing. These 3 files should be what you end up with. And if you want to peek at these files, this is what it looks like. This is the GTF files that you might be familiar with. That seems like a file with a lot of columns. So one trick is to use the capital S. Let's see. S, G, I can't read. Start again. Okay, and now you can see it as columns, and it's much more clear. So I think you might have gone over GTF files before, but if you haven't, I guess I'll go over them really quickly on Thursday. I'll figure out if you've gone over them by going through the past files. And I'll answer Joanna's question in a second. But basically a GTF file, you have your chromosome position, you have the database that the annotation comes from, you have the type of annotation it is, your start and end positions, and then a bunch of other information like the ID of the gene. Okay. Q to get out of less. So if we're going to change the different output folders, let's go to my BI20 test. Okay, so here is our download references param input file, okay? And our first input was the output path for all the files. But if we want to put some files in one path and some files in another path, we can create another variable called outPath2 equals $2. Now this file is going to take 2 inputs instead of 1. So let's say we want to put our FASTA file in one path, and then, whoops, our GTF files in another path. So now we've used this variable out path, the first input, to direct the FASTA file to one place, and then the second variable pushes the GTF files to another place. So I'm going to Control X, yes. Let's double check that our changes have been propagated. So that's how you would change the file. If you're going to now use this, you have your path 1, and then you have your path 2. These paths have to have already existed, so it's not going to create a new path for you. Like you can't put in a path that doesn't exist yet. You have to make a path and then input that as the second variable. But that's how you would do it. And that's about all I have for today. Next class, we're going to go over creating indexed references. We're going to align the reads to that reference. And then we're going to do some visualization. But since some of those steps take a long time, I'm just going to write the scripts, run them, and have them set up. We're not going to, we can submit them in class, but we're not going to use your outputs for the visualization because I don't know how long it will take for all of that to be done. So I'm going to have that ready for Thursday. And yeah, that's all for today. And it's about 4 o'clock. So if there's any outstanding questions, I'll take them now. But other than that, I'll see you on Thursday.